I'm going to share my screen now.
That's it.
Okay.
I ask everyone to turn off the microphone.
And all right. Let me just. Perfect. Okay, so hello everyone. My name is Natali Delorme, and I am an aquaculture scientist at Cawthron, at the Cawthron Institute, focusing on shellfish stress and health. Before I start the seminar, I wanted to thank Marcelo for reaching out to me and inviting me to this club. It is a pleasure being able to interact with lots of scientists and a lot of you around the world. Sorry for the time difference. So yes, thank you, Marcelo. Today, I will present an overview of the research that I've been involved in during the last 5 to 6 years, more specifically about the stress response in the New Zealand Greenshell mussel, which is the mussel that you see here. Here in the picture, you can see rope with little tiny baby mussels attached to it. So that's a really cool picture. As I mentioned before, I work at the Cawthron Institute. This institute is the largest independent science organization in New Zealand, and we recently celebrated 100 years since it was established. The institute is located at the top of the South Island in Nelson City, here. A small city, but with a lot of tourist attractions. Cawthron Institute has different locations around Nelson, but I'm based at the Cawthron Aquaculture Park, which is here, what you see here in the picture at the bottom. The park is about 20 kilometers from Nelson City, which is about a 15-minute drive, 20-minute drive from the city center. At the park, we have different facilities, such as the Finfish Research Center that you can see here. The main building is the shellfish hatchery, which includes a biosecure zone with all the wet and dry laboratories. And this building also includes the main offices of the staff that work on site. We also have a shellfish nursery, smaller here in the circle. And also a recently founded National Algae Research Center to do some aquaculture, algae aquaculture and production. At the park, we also have a few different tenants from the aquaculture industry and also the education sector. We have a mussel hatchery and an oyster hatchery, with a new oyster nursery being built at the moment. And we also have on site the aquaculture facilities of the Nelson Marlborough Institute of Technology. So as you can see on site, we have a big diversity of entities, making collaboration with industry and teachers and students a lot easier. One of the big research platforms that we have in the aquaculture group is the Shellfish Aquaculture Platform. This platform is funded by the New Zealand government, and its main objective is to enable the aquaculture industry in New Zealand. Within the platform, we have three main impact areas, which are growing the existing industries, including Greenshell mussel, we call it GSM, and Pacific oysters. The second impact is enabling emerging species, such as the geoduck clams, which is what you can see here in the picture. Oysters, flat oysters, scallops, abalone, and other clams. And the third impact is securing the shellfish aquaculture production, investigating tool developments and potential risks to New Zealand aquaculture. And the key guiding principle for the platform is to create opportunities to connect research, people, and capability within the platform and with our collaborators and stakeholders. The work that I mainly do within the platform excuse me, falls into impact 1 and 3, enabling the GSM mussel industry, and also being part of the genetic improvement of GSM, as well as developing stress and health tools and investigating the potential risks to New Zealand aquaculture, particularly from climate change. Even though it is not my main focus, I have also done some work within Impact 2 by applying stress and health tools developed within Impact 3 to better understand the biology of new species, for example, in Guida clams. So at Cawthron, to study shellfish stress, we use different tools from oxidative stress, physiological markers, flow cytometry, omics. We do a lot of this work in collaboration with other institutions here in New Zealand, such as University of Otago, University of Auckland, Auckland University of Technology, and the Malagan Institute of Medical Research. We evaluate stress in shellfish exposed to different stressors, such as changes in environmental condition, anthropogenic factors such as manipulation or transport of the shellfish, and exposure to pathogens. This is a diagram that shows specifically for the greenshell mussel the different tools that we use to assess stress and health of different life stages, from larvae to adult mussels. As you can see here, it is a huge range of tools. Some of these tools are used in the whole animal, while for others, we need lethal or non-lethal tissue samples. The white circles, I'm just going to zoom here so I can show you better. So the white circles represent the tools that can be applied to whole individuals, while the black circles require some sort of sampling, either tissue or hemolymph. As an example, I'm showing you the tools that we use on mussel juveniles because it's my main focus of research. And you can see that in this life stage, we measure growth, respiration, ingestion, gaping, viability, heart rate, HSP70 gene expression, oxidative damage, and antioxidant markers. Also histology, omics, so yeah, a whole range of tools. And we also assess survival, of course. So another important facility for us at Cawthron in order to perform disease-related work is the PC2 facility. Having this building allows us to do all of our pathogen work in a contained, secure facility. Here we have several rooms of PC1 and PC2 level with dry laboratories as well, and including a pathogen culture lab. So even though during my master's and PhD I focus on sea urchin thermal biology, my role at Cawthron is to investigate shellfish stress, how stressors affect the animals, and what the mechanisms of response are. I also focus my research on tool development, what type of tools we can use in the lab and in the field, and also combining shellfish stress and tool development to look at spat performance. I have also done a little bit of work looking at selective breeding of thermal tolerance, and I also support other research projects within and outside of the shellfish platform that have a stress and health component to them. So in this aspect, I have been able to work on Chinook salmon, for example, Guida clams, and other clams and snails. So why is my focus on mussel spat, mussel juveniles? Well, in New Zealand, around 80% of the mussel industry relies on wild-caught spat from a place located in the North Island, 90-mile beach, that you can see here along the west coast of the North Island. Here, the spat arrives to the beach attached to seaweed, as you can see here on this picture here. The spat is collected and transported around New Zealand for their final destination. Transport can take up to 72 hours depending on where the spat is being transported to. So now the problem is that 85% of the seeded spat in the farms is lost in the first few months after seeding. But it is still unknown what is the driving of these spat losses. So part of my role, together with other colleagues, is to try to understand the drivers of this poor retention and also reduce spat stress, for example, during transport. I'm gonna have a sip of water. One of the papers that we published in 2021 that got Marcelo's attention actually was to determine how air exposure and humidity levels during air exposure influence the stress response and recovery of the spat when they are put back in the water. We studied 3 times of air exposure, 1 hour, 5 hours, and 20 hours, and 3 humidity levels, low, mid, and high. What we found in this study is that relative humidity during air exposure is critical for survival of the spat, and that oxidative damage during reoxygenation was reduced with increasing relative humidity, even in the longest immersion time. Also, the antioxidant activity didn't increase during immersion in all humidity treatments, which made us think that these mussel species may have a reduced POS strategy to cope with the reoxygenation stress. So the message from this research, and we say this to industry, is that the spat should be transported in a high humidity environment, especially if transported for a long time, in order to maintain a good physiological condition during and after transport. Another research area that I have been working on is immersion hardening of the spat, making spat stronger by preconditioning the spat before transport. In this study, we used immersion time as the inducer of a mild stress prior to simulated transport. We used a short immersion of 1 hour, a longer immersion of 5 hours, and a cyclic immersion of 1 hour immersion plus a 1 hour immersion for 3 cycles. We found that by preconditioning the spat, we were able to stimulate several antioxidants that can help reduce cellular damage in mussels that experience transport and subsequent immersion in seawater. This is a preliminary trial, but it shows that preconditioning a spat could be a potential treatment to increase stress tolerance and reoxygenation stress. It is also important to note that preconditioning and hardening need careful management as to not overstress spat, which could lead to higher energy demands. We have also investigated the kinetics of the stress response in the greenshell mussel when exposed to an acute thermal challenge. In this case, as an example, we had, I show you this graph, we had control mussels maintained at 16 degrees, and also a group of mussels that were exposed to a 1 hour heat shock. We sampled hemolymph and gill tissue from those mussels at different time points during the heat shock and during the recovery time. And we observed that different markers responded differently depending on the time of sampling. For example, we saw that a superoxide radical in blood cells responds very quickly to the stress, while HSP70 gene expression in gills remains elevated almost until a day post heat shock. And that SOD activity, superoxide dismutase activity, and total antioxidant capacity decreased after the heat shock. This graph is a very brief summary of the main findings, but we looked, in this study, we looked at 5 different genes, 4 antioxidants, 3 oxidative damage markers. We looked at 3 hemolymph parameters. We also looked at respiration rate and gaping behavior of these mussels. So a lot of work on this research. Greenshell mussel thermal tolerance is a major area of research for us. In this study, we determined the heat tolerance of spat coming from different selectively bred families and at different ages, from 6 weeks old spat, post-fertilization, to 52 weeks old individuals, so 1 year old. We used a heat and recovery bioassay to determine median lethal temperature, so LT50. exposing the mussels to a 3-hour heat challenge at a range of temperatures, plus a recovery time before assessing survival. We found that different families had different heat tolerance, and that this difference in thermal tolerance is more marked at an older age. We also found that early juveniles are more tolerant to heat compared to sub-adult mussels, the 1-year-old mussels. We determine survival in small mussels by using the fast green method. This method is basically you expose the mussels to a freshwater bath to induce valve closure in live individuals. Then you expose the spot to a freshwater bath containing the green staining. And if the mussels are unable to close their shells due to the freshwater and osmotic shock, they will be stained and classified as dead. I am explaining this method because of the picture that you can see here. Something that we found, and it was quite a surprise, we found that in every family that we studied, even in the most tolerant families, the larger mussels were the ones that were more affected by temperature and dying more. We found that this difference was more evident and statistically significant by the time the mussels were 10 weeks old, as you can see here in this graph, which is very interesting, something that we have never seen before. And it's also interesting because the industry likes fast-growing mussels, so it's interesting to see this. Also, in this trial, we measure HSP70 gene expression in gill tissue of the 52-week-old mussels after being exposed to a 3-hour heat challenge at 31 degrees and then put back in recovery at around 19 degrees for 1 hour. We found that control mussels not exposed to the heat shock had a very similar expression level across all families. But if mussels are exposed to a heat shock, the gene expression of HSP70 increases significantly compared to control mussels of the same family. We also found that the different families have different capacity to upregulate HSP70 when exposed to a heat shock. Interestingly, here you can see that family C was one of the families with the lowest upregulation, but it is also the one that showed lowest survival and thermal tolerance. So that's very interesting to see. Also, when plotting the mean values of LT50 obtained for each family against the mean HSP70 expression for each family, we can see a positive correlation between these two variables, which we found it was really nice to see, especially for the first time in the species. This is another example of a thermal tolerance study done at Cawthron. This study is led by my colleague and now club member Jessica Eriksson. She's probably sleeping now at this time. She investigated the effect of different type of thermal stress on 3 different families of mussels. I'm just going to, yeah. So Jess used different thermal challenges, one where she maintained mussels at a controlled temperature, a ramp and hold treatment where the temperature was increased up to 26 degrees and left there, and a ramp and recover treatment where the temperature was increased up to 26 degrees, but then it was decreased to ambient. She had different sampling points during these treatments, and she evaluated daily survival. What Jess and the team found here is that there is again a differential response among families, and then the transcriptome and microbiome are affected by the thermal challenge and also by the family background. You can see here the survival curves and a little bit of a summary of what this study performed. So as a summary of all the work that we have done over the years about thermal tolerance on greenshell mussel at Cawthron, we have produced a Cawthron report. This is an internal report that we produce, but it can also be shared outside of the institute. This poster is included in the report and shows all the physiological responses that have been investigated under the different lab studies that we have done, such as acute studies, subchronic, and chronic stress studies. And this is another poster that is part of the report, and it summarizes the implications for marine farming. Remember that a lot of the work that we do enables the industry and informs industry, the aquaculture industry. If you're interested in reading this report, let me know and I can send you a copy. So another important piece of work that I'm currently working on is about transgenerational plasticity. For this trial, we use the same 6 selectively bred families that I showed you before in the previous slides, but now these mussels were 2 years old. We conditioned the adult mussels at 2 different temperatures during gametogenesis. We conditioned them at 17 and 22 degrees for 3 months. We sampled them during conditioning to assess overall health using histology, and we also sampled gonad tissue to determine some markers like HSP70, gene expression, and oxidative distress, and antioxidant markers, as well as lipid profile. After conditioning, we induced these mussels to spawn. Unfortunately, the mussels that were conditioned at 22 degrees didn't spawn, so we had to continue only with gametes from the 17-degree mussels. So with these gametes, we created 4 new families based on best crosses of male and female factors, and the embryos were reared at 17 and 22 degrees. We rear the larvae using these bullet-like tanks called cuddles, which is something that Cawthron developed, and it's called cuddle after Cawthron ultra-density larval rearing system, which can hold 1 to 1.5 million larvae in a flow-through system and in about 2 liters of water. So it's a very intense culture, high-density culture, but it works really well. We also ran an early development assay to assess the first 48 hours of development, and we measured larval growth throughout the larval rearing. Now, at the moment, we finished culturing the larvae in these tanks, and we have now a spat settling in these buckets with rope. You can see the rope inside. At the moment, they are about 400 microns. So yeah, they are there growing, and I hope that I can run a heat wave challenge plus a pathogen infection in the near future. So fingers crossed that everything goes well with this mussel spat. As I mentioned before, part of the things that I do at Cawthron is tool development. We have been investigating alternative ways to determine spat health. In this study, we optimize staining that can detect levels of reactive oxygen species in tissue of live spat. Using this probe and image analysis, we are now able to detect different levels of stress in a small and translucent mussel spat. This probe is also fixable, so we can fix the spat after exposure if we want to do so, which can allow for field sampling and transport of the samples to the laboratory. We are also working on the immunodetection of HSP70 in mussel tissue. Here you can see some examples of gill tissue, gonads, and food from mussels exposed to different temperatures. This is very early stages, but we have optimized the protocols for this immunodetection, and we are now exploring some different options to quantify the signal using image analysis. The quantification method most likely will be tissue specific, as we found it is the best way to quantify the signal coming in this all these different tissues. So a lot of the stress tools that we use require hemolymph, and therefore we use them mostly in adult mussels because hemolymph collection is very easy in adult mussels. However, because of the size of the spot, there was limitation, and we couldn't use tools that use hemolymph. So we have optimized a method for hemolymph collection. I will just zoom in here, where we clip part of the shell of the spot without damaging the tissues, and we hold it against a bright light, and we can easily see the adductor muscle of the little spot, so we can actually insert the needle and extract hemolymph. So with this method, we can now collect about 100 to 300 microliters of hemolymph from the spot, as small as 13 millimeters. So this is something really important for us. This new method has allowed us to collect hemolymph from spot and use it to test the stress markers using flow cytometry and flow cytometry devices, and also total antioxidant capacity using this little device here, which uses a liquid sample, so hemolymph in this case. So after developing the new method for hemolymph extraction in a spot, we went to the field and tested the method in a moving boat. Not the same as doing it in the lab, nice and quiet. Here, we were successful at collecting hemolymph from the spot in the field, and also using the TAC device, the little thing that I just showed you, to measure antioxidant activity in the spot that was going through the process of interseeding. So what is interseeding? This is the process where smaller spot that is now bigger and too dense, like you see here in this rope, is taken from the rope and reseeded onto new rope at a lower density. A process that is very mechanical and that is very stressful for the mussels, and it causes a lot of mortality after it's done, especially during the warm season, during summer. I just wanted to highlight that even though we are not an education institution, we do collaborate with universities supervising master's and PhD students. Here are some examples of some of the studies that were led by student projects. And as I said, we're always happy to host students and collaborate with researchers and with students. So if you're interested in visiting us or want to collaborate, just get in touch. As an aside note, and as an associate editor, I wanted to let you know that the New Zealand Journal of Marine and Freshwater Research publishes papers on all aspects of aquatic science. There are no publication charges and free color printing. So, yeah. Finally, I would like to acknowledge all the work of other Cawthron scientists and technical staff, external collaborators, and our funding agency, the New Zealand Ministry of Business, Innovation and Employment, for all the support to carry out all the research presented here today. All research has been a team effort, so by no means is all me alone. So just wanted to recognize that. And that thank you to you all for listening, and I hope you enjoyed the talk. Is everyone there?
Oh, thank you. Thank you.
Thank you. I hope you enjoy that. It's early here, so my brain is still waking up. So, so do you have any questions for me? Who wants to come to New Zealand and visit us?
Yeah, I would just want to thank you, Natali. Thank you everybody to attend this meeting. So we are trying to set up this seminar club. So the whole idea is for people to interact, yeah, because we have people now from 16 countries, and it keeps growing. So I will open for people to ask questions. It's very nice presentation. I was impressed by the quality of your research facilities. Really nice.
Thank
Okay,
you. Yeah.
people, ask questions.
Questions. Okay. Natali, it was a really very nice presentation. I know your work, really. It happens to read your last papers. And really is very interesting work. And I can say that we run common experiments. We are working on the Mytilus galvalucialis. Probably you know.
Yeah, I saw the presentation as well. Yeah.
Yeah. And really, we have such problems with spots. And during last years, because of temperature, we have mass mortalities of mussels. And we try to see how we can improve thermotolerance. That's really a question for me.
Yeah.
And but I would like to ask you. Probably this species is submerged, is living into the water. So this species has an experience with air exposure like the wild mussels.
Yeah.
Okay. So because I heard that you have problem with transportation.
Yes. So we think that that's one of the main stressors, because it's a long time and the conditions may not be the best for collection.
Yeah. For me, it means that this species can tolerate exposure during transportation. Did you measure any product during transportation, for example?
No, unfortunately, we haven't been able to test any markers during the real transportation of the mussels, because it's just a little bit difficult to do so when we are so far away and it's a very far location. But we are definitely is something that we are considering. The mussels, in some times of the year, we have seen and we have heard from even the industry people that the spot during transportation, it behaves differently during winter times and more warmer months.
Yeah.
So also that we also something that we don't know is the condition of the spot arriving to the beach. It has it been eating lately, and all those questions that we are still so they are unknowns for us, and that could also influence the transport. So yeah, we have seen very good transport spot coming really well after those transport and being seated and performing really well, but also some that arrived and you can smell. You know, when you say you smelling dead mussels, you can smell that. So and also because it comes attached in seaweed and all that, it becomes, the longer the transport, it's the more anoxic it starts becoming, all that, because they bag them and they transport them in bags. So yeah, they can stand some transportation, but we have seen that they get very stressed during that.
Okay. And I don't know, have you determined any time the scope for growth rate?
Oh, yes. For the little mussels, we haven't done that for the spat. But for...
This really is very important, you know.
Yeah. Yeah.
The very good indice of the ability of individuals to assimilate the food, take energy. So it's very important, I mean, in the end of this experiment, to see whether you keep a scope for growth rate is keeping or not, is maintaining or not.
Yes, I know that some of my colleagues have, before even I arrived here to Cawthron, they were doing a scope for growth, but in the adult mussels. So these the big mussels. But since I arrived, is when we started actually looking into the health of the spat. Before it was mainly looking at retention and counting what the spat losses and things like that. But so it's only been recently that we have been focusing most on the health of the little babies, and looking at markers.
Okay, that's for the moment, Marcelo.
Okay, thank you. Thank you for your questions, Basili. I have a question. I got really interested in the probe you said for the small mussels, the baby mussels. So
Yes.
I was wondering if we could use this probe to study tardigrades. Because it looks like the same principle, yeah. Is it something specific for yours, for mussels, or can we use for other, like, totally different animals?
Yes, definitely. This is a probe that you buy in Sigma, or I can send you the details. I don't want to say anything here. No, it's called Telrox Green. Yes, and it's a ready to use probe. You just need to dilute it to and optimize it for what you want for your species. I guess.
Yeah. I think I saw a paper from you about this. Can you send it to us in the group? So it'll be nice.
Oh, sure. Yeah, sure. I can do that. Yes.
Because it's very nice. So you can classify dead or alive. It's good. Yeah, it's very practical.
Yeah.
Okay.
Yes. So you mean the fast green method, or the ROS, the translucent?
No, the fast green.
And okay.
Yeah. Actually, you showed two assays.
Yes.
Yeah, the ROS.
And they happen to be both green. Yeah.
Yeah, which is possibly the probe that everybody uses. Yeah.
Yes.
Possibly. Yeah. But the green one, I got really interested because it shows dead or alive. So it's
it's very easy. Yeah, it's very easy to identify. However, it has a caveat that if the mussel is moribund and it's alive but not able to close their shells, they will get staining, and you will classify them as dead. So I like to call it a viability test, because it is either dead or non-viable, because it's gonna die anyway. It's wide gaping and it's not responding to the osmotic shock. So yeah, it's more like a viability dye.
Just one more question. What is the principle for this dye? How this works?
Oh, it's very simple. It's the dye just get uptaken by the tissue. I believe it's almost close to a food dye kind of thing, but more analytical. So yeah, it's very simple. It just stains the tissues because the tissues enter in contact with the dye. It's very simple.
Okay, thank you. Okay, let's open for other people. Okay, Alexander.
Yeah. Okay. Thank you. Thank you very much for very, very interesting report, and the presentation is very useful. And I'd like to thank you very much about for the lot of practical approach that you presented here. Because maybe it was very interesting for me. My research object is really close to your lab. Here in Russia, we also have similar problems with spat viability, because you have problems with transportation, and our spat was For the most, in most cases, about 90% of spat was brought from the Mediterranean Sea. We bought it from France, and there are differences between salinity levels in the Black Sea and the Mediterranean Sea. It's the salinity is twice lower in the Black Sea, and therefore we have huge problems with viability of the spat. And now we are like in the start, in the beginning of the long way of producing spat here in Crimea for our local farms. And we have also problems that here aquaculture is focused on farming of the Pacific oyster, and naturally the Pacific oyster is not presented in the Black Sea because the salinity is too low for it. And thank you very much for these approaches about the assessment of health state of spat, because here in our lab we do a lot of experiments and investigations of health markers and oxidative capacity in adult mussels, and it's very easy. Or adult mollusks, oysters, but we have a huge problem about how to assess the health markers in the small spat when they are too small to sample hemolymph, for example, individual, of course. And I have some questions about, you mentioned that you use transcriptomic and microbiomic analysis, and which tissues are you use for microbiome analysis, for example, or transcriptome? Do you use hemolymph or like it's total sampling? Which tissue?
For the adult mussels, we use. Thank you, Alexander. That's very interesting to know that you also have the similar problems, and that you are now raising your baby mussels for your local farmers. That's really interesting. So, for regarding your question about what tissues we sample to do transcriptomics and microbiome. For the adult mussels, we have done that in hemolymph and gill tissue. And in baby mussels, we have done that in a whole mussel. So unfortunately, because these are, you have different sizes for the ones that you can sample blood, for example, the 13 millimeters, that would be easy to do with the blood. But the other ones are 1 millimeter. So the spat that comes in the seaweed and arrives to the beach, that's 1 millimeter spat, between 1 or even less sometimes. So for that, we need to actually get a pool of a spat, and we cannot sample tissue. So it's the whole animal and tissue from several animals to actually get something for analysis.
Mm-hmm. Okay, thank you. Because now we are also trying to assess microbiome, and we took gill tissue, but I want to understand which tissue is the best for this analysis.
Yeah,
and
we have,
mm-hmm.
Yes, sorry. We have gill tissue, and it has worked really well. I wasn't able to go into deep detail on how the transcriptome results look for that study that my colleague Jessica was doing and looking into the transcriptome of the mussels, but you can really see distinct patterns using gill tissue and for the microbiome and mm-hmm. the transcriptomics for the hemolymph. Yeah.
Mm-hmm. Thank you very much, and I think that we will be in touch.
Oh yeah, definitely.
I have a lot of questions, but they are specific, and I don't want to bother other participants because they're like not linked to redox biology. So thank you very much. It was very, very cute report. Thank you.
Thank you. And yeah, let's keep in touch and see what we can do.
Okay, thank you.
Okay, more questions.
about reproduction. I see one here in the chat.
Okay.
Nice presentation. Could you tell us a bit about reproduction? What aspect of reproduction?
Without the microphone, Elida, exactly what you want to hear. While she writes the rest of the question, I would like to incite Giancarlo to think about hormesis and what she's doing. You know, Giancarlo has been working for a long time in hormesis. Yeah, we published a paper a few years ago. Yeah, and exploring this concept in UV. Yeah, so I got very interested in the effect of UV, yeah, in the animals. While Elida, she's so you answer her, and then you think about the effect of UV on your animals.
Yep. So yes. So the question is how parents are selected. Well, the selective breeding program that the mussels that we are using for the research, it's been 20 years longer. So it's been a long time of mussels being selected. At the beginning, the mussels were selected for growth, so the larger mussels. And for selective breeding, you always want to keep some variability, right? You don't want to just mix and cross the larger ones and the better ones. You also want to keep a little bit of the not so good ones to maintain that genetic variability. So yeah, it's been really long, even before I got here. So I think initially was selecting for size. And now what they are doing and what the method of of the ramp and hold and all those methods are being developed because they show individual performance and individual thermotolerance, and we can actually differentiate between families, all with the thinking about climate change and potential selective breeding for thermotolerance. Before the methods of looking at the thermotolerance of the mussels was to do an LT50 kind of approach, but you have it's a population level response when you have one number of LT50 for a whole bunch of animals. But with this method of ramp and holding, we have seen that at 26 degrees kind of is a tipping point for the mussels, and you start seeing a slow death until all the mussels are dead. So we have seen that with this method, actually, we can see individual thermal tolerance response and model it to actually get a difference between the more thermotolerance families to the less thermotolerance families. So I'm guessing that now the industry is starting to think about a little bit more climate change and breeding for resilience. So yeah, I think from selectively breeding for growth, we are starting to move towards a more climate change friendly approach for selective breeding. And the UV question, yes, now I remember, sorry. I saw your face, Marcelo, and I remember the UV question.
Yeah, especially in the big ones, yeah, because they're transparent and, you know.
Yes, it's something that we haven't studied, but definitely there are some studies on UV effects on intertidal species and things like that. So I think it's something that we could definitely look at, but we haven't yet done any studies on that.
Yeah, we just published a paper on the effect of UV and tides on oxidative stress and antioxidant response in mussels. But our mussels the adults are like this, you know,
yeah.
very small, and there's straight from nature. Yeah, we collect everything was collected in nature, in time points, and then brought to the lab. And we found that all the effects by the biochemical responses are all linked to the presence of a certain level of UV radiation from the sun. Yeah. So
very interesting.
Yeah, it modulates. So to have the POS response, it's not just the animal to be outside the water. It is not enough. It needs to have UV too. So it's both.
That's interesting. And that is something that in the lab is very difficult to do sometimes. The multi-stressor studies, they are very, very important. We all know that it's very difficult to do a lot of stressors in the laboratory. So it's great when you can go and see and assess animals from the wild and assess those markers, because you are looking at the whole effect of the real world environment.
Yeah, this is something we are moving to. We're moving from biochemistry, lab biochemistry, to the nature, to the wild. Yeah, and but it's hard because sometimes the weather changes and rain and then you lose everything.
Yeah. I bet.
Yeah. But it's something I think people that work with small animals, translucent animals, you have to think about UV. Yeah, before we wrote this paper in 2020, Giancarlo helped with the part of the ionizing radiation. But in nature, we don't have much of ionizing radiation. It's mostly UV. But for small animals, it's very important. Most people don't care very, very much. Yeah, and in small amounts, it induces an antioxidant response, and in high amounts, it's toxic. So it is the classic hormesis effect. It's very nice. We want to say something about this, Giancarlo, because he studied hormesis like for 15 years. Maybe we will invite you very soon for a talk. Yeah.
Well, yeah, thanks for that. It is something that I thought about in multiple places, and not just UV, right? When you were talking about your data with humidity and the changes, in the past I have worked with dehydration or rehydration hormesis, where mild dehydration can actually end up being very protective. And so sometimes in a situation where relative humidity is manipulated, below 100%, being in the 90s, above 95% but below 100, can trigger this hormetic response, similarly to the hormetic response that we see from UV, that would trigger antioxidants and certain protections that lead to the reduction in oxidative stress. So whenever I see any sort of like relative humidity manipulation below 100%, I'm always interested, because it could trigger that hormesis, just like UV does, right? In my lab currently, we are working with UV hormesis and bees, right? And we're trying to do some things on the field, and that's problematic, literally for the reasons Marcelo was just saying. We just had a big storm last week, and it took most of our nests down because it was, wind and rain and thunderstorms, right? And so we're always trying to play catch up. We're trying to find some relevant UV exposures that we can test in the lab.
Very interesting work.
So let's invite you for the fourth seminar, if you agree. The third one will be from Cubilus from Chile, and you can be the fourth one if you would like to.
Send me a message. We'll put it on the books.
Yeah, okay, that's good. Okay, do we have any question for Natali, Giancarlo specifically on what's?
Marcelo? No, I don't. I mean, it was a great presentation, right? And yeah, very comprehensive. comprehensive and really gave me a good picture of what's going on with the mussels and their responses, right? I thought it was really interesting, this family difference, right? So I guess it's like this generic background difference on the stress response, because it's something that we don't tend to think about. We think the stress response of a species is like the stress response of a species, right? But there's so much that genetics into play with it, and likely, obviously, transcriptomics, right? We see very different Hitchcock 70 responses, and I thought it was like really cool, because normally people don't even chase that genetic difference, right?
Yeah, we definitely are very lucky to be able to work with these selectively bred families. This is a huge. It's a long selection of these families, and being able to have it available for research is invaluable.
Yeah, as I was looking at especially the Hitchcock 70 expression data, I was sort of wondering how much of that is a strong expression versus if they might have different copies, if the different families might have different copies of the gene, which normally allows you to express more, right? But I know it wasn't part of the presentation, but that just came to my mind, right?
Yeah, no, it's something good to consider. Thank you.
Thank you.
Something, Marcelo?
Yes?
Can I ask something to Natali? Okay, thank you very much for the presentation. It was very comprehensive one, and it was a very nice thing to learn what you are doing there with Perna mussels. I'm not working with marine animals actually, but we have seen that several populations of the same species have different reactions, and this result of yours with different reactions in the different families is very, very important and very triggering for our work too. What I would like to learn is how these families were established, and for how long do you keep them in the lab, and which strains did you select to produce these families?
This, well, as I mentioned, the selective breeding program started around 20 years ago. And we are lucky to have, if you remember, at the site, we have some tenants, and this mussel hatchery that is on site, they are the ones that own the families, and they do run selectively breeding runs every 3 years, more or less. And they keep their adult mussels in the farms, and they just keep bringing them when they need them for spawning and to create more babies. So yeah, we have direct access to those. So when we need them and when we are thinking about doing something with them, we just need to be tying perfectly with their selectively bred breeding families, breeding program, when we want to do something with the larvae or the baby mussels. But if we want to work with adult mussels, they keep them in the farms, and they are very nice to us, and we can just ask them for some numbers to do the trials, and yeah. So initially these mussels were starting being selected for growth, and what usually happens is that you see in the wild the animals that you like, just as a phenotypic characteristics. You see them bigger, and they started doing the selective breeding program based on growth. And as I mentioned, now they are trying to move more and understanding a little bit more on the thermal tolerance of these families and whether they can select for resilience, thermal resilience. Yeah, so we are very lucky, as I said, to be able to work with these families, and yeah.
Thank you.
I have another question. Yeah, going back to the studies that I was interested, like a month ago, I would like you to explain this effect of humidity, like the mostly the two extremes, very low humidity and high humidity. How you think the high humidity protects these animals?
To be honest, well, the data of the markers is there, and you see how the low humidity causes more stress and the antioxidant response and all that. But for me, what really was really nice to see when you're running the experiment and you're sampling the mussels is the one when I sample mussels, the juveniles from the high humidity treatment, for example. They were glossy, they were nice, humid. You see them happy. But when I sampled the ones from the low humidity treatment, you could see them, they were crispy, so so they you can see how that could affect a mussel, and with the green shell mussel and the baby mussels, we've seen that when you leave them on the counter, or they gape and they lose a lot of that humidity, internal humidity during that gaping. So that's why we thought the relative humidity of the treatments was very important during transport. I want to make a clarification about the it's not only transport, and we think that transport could be one of the reasons why we are losing so many mussels in the farms, but I'm pretty sure there are many more factors, like maybe temperature of the water when they are seated, food availability, and so on and so on. So it's something that we think it could be part of the problem as well, and that's why we are trying to make the muscles stronger and to stimulate antioxidant response, so they can cope better with conditions during transport, which we think it is one of the factors affecting retention.
Yeah, it's interesting because even in low humidity, they produce lots of antioxidants and they survive. Yeah, but then when the hours are passing by, so this antioxidant defense let's just go down and all falls apart. So these animals they need humidity. Yeah, after a certain time. Yeah, definitely. And it's interesting as well to see, for example, in that it's a completely different study, but in the kinetics of a stress response that I showed, we see that yes, the antioxidant response goes up at some point, but then when the stress is too much, it goes down. And it was for that specific a study that unfortunately I couldn't show all the data. We saw that the gene expression and also the antioxidant activity decreases, and it's downregulated. The genes of the enzymatic enzymes, enzymatic antioxidants, are downregulated, and the activity also it's reduced. So I think there is a point when they try to cope with the stress, but then it's everything is lost. And probably that is related to temperature. Yeah, possibly lower temperature. It may be more favorable for these animals, even in lower humidity. So it's a multifactorial. Yeah, I was reading a couple of papers yesterday discussing this importance of looking at several factors. I mean, in an orderly way, but usually people study temperature and that's it. And in nature, there is no only temperature stress.
Yeah, it's true. For us, of course, as scientists, we've seen a lot of the papers is just temperature, all the older papers, and more and more we realize that we need to start doing the multi-stressor studies. But they are difficult to do. So we can only try to do the best to our abilities.
Yeah, of course. Yeah, we can do the best we can. Yeah. Okay. Any more questions?
Natali, sorry.
Yes.
Did you run some experiments to see what's the best condition regarding temperature, humidity when you transport the sponge?
No, unfortunately, we didn't use a temperature as a factor for the study. Yes, we maintain a constant temperature for the simulated transport, but yeah, it's definitely something that is it must be important, and it must be very important.
And you can see what is the best condition when they are exposed to air. Okay.
Yeah.
But I have another question. I know that lastly, many investigators use hemolymph to examine the health, muscles health, and they apply metabolomics, etc. But all metabolites are excreted to hemolymph. I know that why you don't use tissues as well, because
Yes,
some
we have.
metabolites are excreted in hemolymph, so probably may have artifacts.
Yes, definitely. So some of the studies, for example, the one that I showed, where we use different tools to assess the health of the integration of tools, and we use this Greenshell mussel as a case study. We use the two different tissues, so hemolymph and gill, to see how different they are. And yes, you could see that there is a difference in the metabolites that you find in these two type of samples. So yeah, that's what I particularly investigated some time ago. But then for each particular experiment, depending on what the experiment is for, we try to sample the tissue that we think is best. For example, sometimes we do with the hemolymph extraction, we can do repeated measures on the same animal because it's a non-lethal sampling. So that's a very, very good advantage to actually do that and analyze. So there are some reasons why we use different tissues for the different So yeah, definitely the metabolites change depending on the tissue and the tissue specific response. Yeah.
And we know that the muscles are full of microorganisms. So some of metabolites may be byproducts of microorganism metabolism, and not the muscles.
Oh, yeah. Okay.
Yeah.
That's good. That's interesting. Yeah.
Yeah. Too early, too many questions, Marcelo.
Yeah, that's good. The more questions, the better we learn.
It makes me wake up. No, it's 7:15 already here, so it's all good. Yeah, it's all good. I'm well awake.
Do we have more questions? Ken Story, do we have any questions for Natali? Yeah.
I have no questions. I don't go outdoors very much. And when I do, I tend to sit in a chair and drink coffee. So I watch people doing outdoor research.
Nice.
You may not be able to. Okay, good.
Yeah, okay, thank you. Yes, with a big bag of coffee.
Yeah.
Yeah. Okay, thank you. Is anyone else wants to ask questions? So, if nobody wants to ask questions, I would like to thank you, Natali, for your very nice talk. And you please send to us, to the group, your recent papers. People will like it on PDFs. And I'd like to thank you, everyone who attended the seminar, and the ones that asked questions. Oh, hello, Ken. Yeah. Yeah.
Yes, thank you, Marcelo, and thank you everyone for joining. And yeah.
Yeah. So thank you.
Thank you.
Thank you let's take a picture, everybody. Yeah. Okay. Thank you, everybody.
Okay.
So,